It's time for Dr. Rademaker's Prescription for Transformation. Let's tap into your own authentic genius and live the life you desire. Join Dr. Rademaker for an enlightened conversation about our brain intelligence, our body intelligence, and our energy intelligence. And now, here's your host, Dr. Bart Rademaker.
I'm Dr. Bart Rademaker, and this is Prescription for Your Transformation. Real people, real conversations, and real success. And today's talk is a really interesting talk. It's one of my favorite talks. It's about the stem cells. It's about regenerative medicine, and obviously this crazy time that we live in today, the COVID-19. And so I've already talked about this in the past. A lot has happened since then. I've done a whole ton of research. I mean, this amounts to about a 20th of all the research I've been doing since I last talked to you guys. And so we actually do live in extraordinary times. It's a very stressful time for many people, but it's also that time where we need to begin to assume that responsibility. With great crises, with great power comes great responsibility. And today we have that responsibility as medical professionals and just entrepreneurs, business people, politicians, you name it. Every single person has that responsibility to start making some really smart choices, because it's a global impact. It's affecting everyone. But today I want to talk about a whole bunch of different things. But first I want to put it in perspective. I mean, pandemics are not new. They go back way to recorded history. And clearly back in the 1300s, you had the bubonic plague. Interestingly enough, that was a plague that was spread mainly by rats. And now we have a flying rat, I believe, as the source of this virus. 200 million people died, at least recorded. I mean, that's at least what we know of. But then fast forward to 1918, and we had the Spanish flu. Today everybody's really upset because we call it the Chinese flu. But then again, in 1980, it was the Spanish flu, because that was the origin of where it came from. And so probably in estimation, about 40 million people died at that time. Fast forward to 2009, and I don't even recall this one, but this was the pandemic that we had, the H1N1 virus that actually started in the United States. It wasn't called the American flu, though. It was also called the swine flu. And so what's relevant today is the importance in understanding, because there's so much fear, because there's so much uncertainty as to the impact that this is going to have on people's lives, what are the solutions? And so some of the solutions that people have already investigated, there are trials out there currently, with antibiotics, also with NK cells, which are natural killer cells. Great company, Cellularity, owned by Hariri and Diamantis, creating these lymphocytes that actually recognize foreign cells in the body and actually go for them. So it's really exciting that they have this ability to do that. But we're also looking at immunoglobulin studies, at IL-6, which is part of what's called that cytokine storm. And then, of course, stem cells. I mean, they're popping up all over the place. And that's one of the reasons I wanted to talk about regenerative medicine today. I wanted to talk about the COVID-19, how it can fit in, how it's a potential opportunity there to treat people. And then really begin to look at, for the consumer and doctors as well, but both are important, is to understand the science behind regenerative medicine, some of the strategies and trials that are out there, but also that responsibility and integrity. As I mentioned earlier, in this crisis, with great power comes great responsibility, and we need to harness this really well. And so for today, I want to start off with a real quick disclaimer. I'm not making any medical claims. Particularly, I'm honoring the FDA and their requirements. As physicians, they don't want us to make claims. They don't want to have us use testimonials. I have 10 years of experience in this field, and having treated patients, and I'm not going to be referring to them. What I will do is I'll refer to any of the peer-reviewed clinical trials so that you can decide for yourself the relevance of the information that I'm giving you. As with anything, I love the phrase trust but verify. This information that I'm going to give you, go ahead and verify that. I'm also going to supply different links that you can look at. One link, for example, is my own website, where if you sign up, I'll shoot you some of the papers that I've and my team and myself has summarized, so that you can easily understand what these trials are saying. You know, I look at these scientific reports and I get confused half the time. So I need my scientists to help me understand what it is that they're actually saying. So what is regenerative medicine? I mean, you've heard things like stem cells, embryonic cells, exosomes, miracle cells. Look, it's a lot more than just the cells. And the problem that we have in this field, there's a lot of confusion. There's a lot of misrepresentation about what it can do, preying on the hopes and the desperateness of other people. And as Gottlieb from the FDA has mentioned, I think it was last year, you know, there's a lot of false actors out there. There's a lot of opportunities out there. We see this right now with COVID-19, actually. All these people who are, you know, trying to sell this PPE and actually they don't have it. And so a lot of opportunism out there. And there was this one store in Canada where this realtor decided to buy up, you know, all the hand sanitizers and then selling it for astronomical prices, which is just wrong. So what I want to bring up today is conceptually understand two things. Is that what is that healing potential? Essential in everything that we look at when it's regenerative medicine, as well as this is a paradigm shift in how we get to treat patients. And this is just a major shift in our understanding and our ability and potential to heal our patients. And interesting enough, you go back to 300, 400 BC, and this is relevant because the whole concept of regenerative medicine is not new. The whole potential, the whole idea of our bodies able to heal itself, that potential within us, is not a new one. And so Hippocrates actually very clearly mentioned that there are natural forces within us that are the true healers of disease. And then you have Aristotle shortly after that, about 300 to 400 BC as well, who's in his works of natural history is talking about or describing how salamanders are able to regenerate their limbs, and actually how deers can regenerate their antlers. So talking about the paradigm shift, and what do I mean by that? It's a shift in our knowledge, and it's a shift in our ability to do things. And what's exciting is that, and as I'll explain a little bit later on, there's really three revolutions that are going on simultaneously right now that totally and dramatically will change our lives. It started off about 20, 30 years ago with a whole wellness revolution, and really focusing on, you know, how do we optimize the nutrition in our body? How do we optimize the toxins or remove the toxins, other micronutrients in our body, so that our body actually does function better? A lot of the science and gimmicks out there and gadgets out there have been looking at that very effectively. But now we also have, so that's the wellness revolution. But we also have the regenerative medicine revolution, where we're getting access, we're using the intelligence of the body again in a different form with cells, with cell therapy. And I'll discuss more of the types of regenerative medicine in a moment. But that's instrumental in how it is that we can heal patients. And so together, you want to look at it from the perspective of, we now have the ability to reduce the burden of disease, improve the quality of life, and then extend life. How cool is that? You know, as doctors, and for so long, and giving doctors the credit, you know, for, you know, all their commitment, all their dedication, and treating patients, but really basically having their hands tied behind the back, because, you know, they're doing the best they can with the resources that they have. Also, with the advancement of technology, I mean, just think about it, it's every 3 to 5 years that our knowledge doubles. And so these poor guys, they're trying to keep up with all the advances, and they just can't. And patients come in, talk to them, and ask them about new developments, and they have no clue about this. And so now with regenerative medicine, that's a whole new arena that we now can give doctors and not just treat the symptoms, but treat what's called the root cause, treat the real underlying problems. And so what is regenerative medicine? Basically, it's a form of bioengineering and molecular biology. Its purpose is to restore form and function. Basically, using cells, using biomaterials and molecules to replace and repair and regenerate cells and tissues. But here's what's also something that a lot of people aren't really quite familiar with, is that it gives us that ability that we've never had before to actually study disease in culture and cells, and also discover and design a drug and new treatment strategies with the use of these stem cells. And so this is quite an innovative approach rather than, trial and error with animal studies and everything else. I think we can much more, in a targeted fashion, be able to understand how the body works and how it is that we can also treat disease. So in fact, regenerative medicine gives us two opportunities. One, so that it addresses the root cause, but also finding situations where we can actually treat the symptoms as well, just like traditional medicine has been doing. So again, no claims. This is an evolving science. Again, the FDA commissioners in this country and other countries have very clearly expressed their concerns in regards to this industry, how a lot of people are actually jumping into this industry and not necessarily in the best interest of our patients. And so they're looking at the entire safety of regenerative medicine, as well as understanding the efficacy before approval is going to be given. And I'll talk a little bit more about some of the concerns that we have as well. So it's important to know the science. It's important to know the regulations. And it's also important to stay on top of it and verify, exactly what is being told to you. And hopefully what I can do is, for anybody who wants that, provide resources so that you can navigate this field with confidence and make smart decisions. In fact, what I'm currently doing, particularly for doctors, is I'm creating a program, a training program. It's basically a 3-week program where doctors can really supercharge their ability to confidently use regenerative medicine in their practice. Again, ensuring that they're compliant with all the types of regulations, because once again, we have to work from a place of integrity and the support of the FDA. So like I said, my purpose here is to educate, to help navigate with confidence and be able to make the right decisions. And hopefully I'll have a program pretty soon for patients as well, so that they really understand the science behind this, know what kind of answers, and know exactly what the FDA's expectations or requirements are of doctors, so that you can make sure that you're making the right decisions, educated decisions for yourself. So it's a shift in the treatment strategies for dysfunction, for loss, for disease, and cancer. So example, I love this example, and this is actually a clinical trial out there where they are replacing scarred cardiac tissue, heart tissue, after infarct, and with new cells. And so by intracardiac injection, the study has actually shown that the size of the infarct has been reduced after a coronary artery bypass. Talk a little bit more about that later on. The other one, and I believe this is Dr. Millman in Michigan, I think it's Michigan, apologies if I got it wrong, but they're studying the ability to grow islet cells that produce insulin. And so just imagine that if a patient with diabetes, type 1 diabetes, lacks these cells that makes insulin, and now they can recreate this, and now the patient doesn't require insulin anymore. I mean, how cool is that? And that is the future. We're not there yet, but this kind of potential is, what's in the horizon. So what's the challenge? Well, the challenge begins with the fact that everybody thinks these are miracle cells. Doctors included, by the way, scientists included as well. The problem is that there really is a paucity of science, but particularly the double-blind, the randomized, and the placebo-controlled studies. A lot of the studies out there are in the laboratory, so these are either cultures or animal studies. Over the years, there's been a significant increase in the clinical trials, particularly in cardiac medicine, orthopedic, arthritis, immune disorders, hepatic disorders, liver disorders, and other conditions. But one of the other problems is the lack of standardization. So what does that mean? The standardization is how the cells are processed, how they're acquired, also the language that's being used. And so it makes it kind of difficult to, when you look at all these different studies, to really be able to make any significant or clear correlations. Sometimes it's very well possible, but other times it's kind of hard, because if everybody has a different technique of doing things, it's really difficult to understand the relevance of their data. And so going back in history a little bit, and this is kind of fun as well. I already talked about Aristotle and Hippocrates from Greece, but in the 8th century, Hesiod was talking about, in his Greek mythology, was talking about the Titan Prometheus. And he stole fire. And this is their mythology. These are their stories. And so by stealing the fire, he was actually punished by Jupiter, and then chained to the Carpathian Mountains. I don't know where those are. And the eagle, Ethon, visited Prometheus every single day and started picking at his liver. And so he was being tortured for 30,000 years. Finally, he was freed up by Jupiter. And so why is this relevant? Because the concept of regeneration was already there. And hepar, which is the root of the word liver, actually means to replace itself. We have the hepatic, the Latin from hepatic, which means liver, and it from Greek, it means to replace itself, to regenerate itself. So again, understanding that regenerative medicine isn't new, but modern regenerative medicine is really 50 years old, basically. Or let me step back a second. So 2 centuries ago, back in the 19th century, 1867 to be exact, a eminent pathologist in Germany, Dr. Conheim, and then later in the 1920s, and Maximo, both described the ability of bone marrow cells to heal wounds or distant wounds, as well as make more blood cells. And so here is already the physical demonstration, or the laboratory demonstration, the clinical demonstration in this case, that we have what's called progenitor cells that lead to regeneration. But in the last 50 years, we started in '68 with the first bone marrow transplant. So this area of medicine has really been ingrained in a lot of therapies today. And so treating blood dyscrasias, leukemias, with bone marrow transplant, transfusion, all these kinds of things. And so that's pretty much mainstream already. So already in '78, we discovered the stem cells in cord blood, and this is relevant because treatments are using cord blood. '96, you had the sheep dolly that was cloned, so that's a whole different arena. We're not going to go there today. But interesting enough, in '09 was the first approved FDA study using embryonic stem cells. And this is important because a lot of clinical trials have begun since then, and they're exploding in many areas. Still predominantly laboratory studies, a lot of clinical studies, and that's relevant for today. And fortunately, FDA and governments worldwide have approved many of the COVID studies, and that's important. When we talk about regenerative medicine, we're also talking about bioengineering. And back in the sort of, I think, 1990s, or 80s perhaps, you had the first engineered tissue transplant to skin. And so again, it's not just the cells, but it's how we bioengineer them. So where did the term stem cell come from? Well, actually, Arnold Kaplan, back in the 90s, was able to culture cells in a particular way and show that they reproduce themselves, and they also differentiated into lineages. And this is basically the definition of stem cells. And we'll go into more detail. But specifically, and I think this is relevant for anyone understanding or wanting to understand conversations about stem cells, we have to look at the in vitro definition, so outside the body. So there's about 5 different things that I picked out. So first of all, there's to define an MSC, and again, verify this information. As with everything, science is evolving. People have different opinions, different ideas. Quite frankly, Arnold Kaplan is wanting to change the name of mesenchymal stem cell, which he coined, to mesenchymal signal cell. Many people actually use the word mesenchymal stromal cell. We'll go into that a little bit later. But the plastic adherence in cultures is one, first identified by Friedenstein. But that these cells can replicate, that they generate progeny, which means that it's a cell lineage that differentiates into actual functional cells. And this is key. That these stem cells persist, and perhaps the most important thing, in my opinion, that is the cells are affected by the environment that they're in. And so why is this relevant? Well, the whole study of epigenetics describes how cell DNA expression or genetic expression is modified without making changes in the DNA itself. And basically, it's the components, the molecules, the environment around the DNA that causes the changes in the expression of the DNA. Interestingly, if you want this fact, is that we have about 22,000 genes. I think there's about a 97 similarity with the Anopheles fly. Correct me if I'm wrong, but we can't explain the difference between the fly and us by just those 22,000 genes. And we can explain it by the fact that there's 2 million switches. So with those 2 million switches, all the different combinations of gene expression then explains the variety in species. So getting back to this idea of environment, I think is key, and I'll talk a little bit more about that in a little bit. But what's key is that as we take these cells, and let's call them MSCs, or mesenchymal stromal cells, I'm not going to use the word stem cells because that's a term that we can't be really using because it's not the appropriate term. It really doesn't define what it is that we're using. I'll also be using the term cell therapy as well. So putting all these cells in order of potentiality. So you begin with what's called the fertilized egg. That's when the sperm connects with the egg, and then you get what's called the zygote. And as soon as that begins to divide, I have to watch my time here. As soon as that divides, then we've got the embryo. And so the zygote is totipotent, so it can create any kind of organism. But the embryo, if you take those cells out, they are what's called pluripotent. So they can deliver all the cell lineages, but they can't create an organism. So within about 7 to 8 days, you get what's called the blastocyst. And again, this is relevant because we have to understand where we get these cells from. But the blastocyst forms the fetus and the trophoblast, which becomes the placenta. And later on, you'll see where we can actually get these MSCs, both from the placenta and the umbilical cord. The fetus becomes, or is formed within about 8 weeks. Then the umbilical cord, so at first the fetus is fed by the amnion, which is the fluid around the fetus. And then you have the umbilical cord that ultimately forms. And at birth, this is a very simple piece of tissue that measures about 40 to 60 centimeters, and its width or girth is about 1 to 2 centimeters. It has 3 vessels, a vein and two arteries. And within it, you have what's called the Wharton's jelly. And so you'll hear about people talking about the Wharton's jelly, and this is key. because the Wharton's jelly is an extracellular matrix. So it's that matrix around cells, it's that connective tissues. But it's considered the simplest form of human connective tissue. Why? Because there's no nerves in it, there's no lymphatic tissue. It's predominantly made out of collagen, hyaluronan, sulfatic glycosaminoglycans, I think I said that right, the fusible protein, and some cells. And so when you do cross-section of the umbilical cord, you'll see that there's a very thin epithelial layer of amnion, and then you have the subamnion zone, the intermediate zone, and then finally the perivascular zone. And in the processing, when I talked about this lack of standardization in the processing, people are processing the umbilical cord in different ways. And by the way, this is where we're getting a lot of the cells, or the allogeneic cells. And so depending on what portion of the umbilical cord you're going to get different types of cells. And so what they're finding is that 45% of the cells are in the perivascular zone. And so I've asked different companies, how do they do that? How do they get the cells? What's the process? No, they don't tell. So when we get the MSCs, from Wharton's jelly, this signifies really a milestone in potential therapeutics. Why? Well, I started with cell therapy about 10 years ago. And the process that I was utilizing, what many people were utilizing then, was either bone marrow or adipose, fat tissue. The access to MSCs from the umbilical cord is amazing because it's discarded tissue, it's non-controversial, it's non-invasive, so therefore it's painless. It has an extremely high yield of cells. The cells are homogeneous and they proliferate or replicate very well. So there's this amazing expansion potential. And so when we're talking about regenerative medicine, we have to keep three things into our minds, or into account in relation to all the conversations. First of all, it's the in vitro and the in vivo situations. They're very, very different. The environments are different. So we can only make assumptions and best correlations between how a cell is expressing itself in culture versus how they express itself in the human body. The environment is simply different. I mentioned earlier that it's not about the cells, but it's about the paracrine effect. And so the understanding today, contrary to what it was before, was that these cells, when they're given to in therapeutic forms, their effect is predominantly a paracrine effect. So it's where the cells release all these different factors locally compared to the endocrine, which is distant. So local deposit of different products then induce the tissue into different changes, particularly the stem cells. Or it's the cell contact that induces the changes as well. So it's contrary to what many people believe, particularly the consumer, is that the stem cells will replicate in the body and therefore do all these things. There are a variety of different studies that suggest that within about a day or two, pretty much 90 to 95% of those cells are gone. But then again, you have other people that say, well, they replicate and you get millions and billions of cells over a short period of time. That's not true. The other part, and particularly in designing treatment strategies, designing different types of drugs in regenerative medicine, it's what's called the induced, or is a pluripotent cell, or the induced pluripotent cell. So the pluripotent cells, these are cells acquired from the embryo. These are donated embryos, and so there's no controversy about that. But then you also have the induced pluripotent cells. And so sometimes you'll hear people talking about iPSC, which means they take a somatic cell, a skin cell, for example, and they induce that with a genetic induction or inducing genes. And what that does is that it undifferentiates into then a pluripotent cell that can create all cell lines. Another way is you can take a secondary oocyte and introduce the nucleus of a somatic cell into that. So it's a different type of induced pluripotent cell. But again, these are valuable tools in medicine to be able to design drugs and treatment strategies as well. So we talk a lot about the adult cells, and these are tissue-specific cells. So they differentiate into very specific functional lines. So it's not like they can change or differentiate into any kind of cell. So say, for example, you take the bone marrow aspirates, and they can go into bone. They can turn into cartilage or even fat cells, but they can't turn into nerve cells or other types of cells. But then you also have neuromesenchymal stromal cells, which are specific along the nerve lines. So where do we find these? We find them in the walls of small blood vessels and capillaries. And this is what's really neat about what I've been learning, is that the stem cells actually live in what's called a niche. It's that niche that has a specific microenvironment that stimulates and sustains the replication of the stem cells. So basically, this is a protective space where the stem cells reside, and they replicate. And what happens then is that as soon as the cell is out of that niche, then it begins to differentiate, and then it's called the MSC, or the progenitor cells. They replicate several times, to a limited number of replications that they then undergo, and then finally develop into a very specific functional line. Now, we talk about pericytes. Pericytes are cells also in the capillaries in the basement membrane. So that's basically in the wall of the capillary. They are known to regulate blood flow. And some believe that the pericyte is the actual niche for the stem cells. So research is in the lab. It's clinical. And what we're discovering, and again, showing you papers, and these are what papers are suggesting, the opportunity, the possibility of therapeutics include immunomodulation, anti-inflammatory, vasculogenesis, making new blood vessels, cell replacement, scar removal, and antimicrobial, and also in the case of COVID, stopping the cytokine storm. I'll talk a little bit about that in a moment. So I've got a couple of studies here in 2006. There's Kirkatovsky, looking at acute and chronic renal failure models, an animal model where they take the MSCs and they're able to recover renal function and the survival of the animal. It's phenomenal. And acute renal failure, chronic renal failure is a big issue, and it'll be exciting when the time comes. I should say it that way. When the time comes that we can treat people with chronic renal failure and get them off dialysis. Then there's another study looking at the therapeutic effects of the angiogenetic cytokine and stem cell mobilization in patients with cardiac disease. So this was actually an infarct model in animals, so it wasn't real people, but really showed how stem cells were mobilized and how vasculogenesis was mobilized to restore blood flow to a heart tissue that was ischemic at the time. Then there's to follow up on that one, in 16, Anastasiadis, they had 11 patients. This was a clinical trial with open heart surgery and coronary bypass, and they got induced MSCs and were able to reduce the infarct size by about 40%, with significant improvement in quality of life. There are other studies out there, Lyme's disease, MS. There's one with 2 patients, one with 59 patients, clearly in showing improvement in patients. I'll have a couple of studies on arthritis, disc problems, and bone healing in diabetics, all showing at different types of therapeutic potential. And so you can decide for yourself as you read them, how this can relate to you. Pulmonary disease in particular, in the times that we find ourselves now, is relevant in a couple of studies that are showing coronary pulmonary disease, which is fibrosis, interstitial problems, as well as the H9N2 avian influenza with all the tissue edema, which is a problem that the COVID is experiencing as well for patients with the COVID, and shows remarkable improvement with those. There are also studies out there, and I'll share some links with you as well in regards to clinical trials in China that are demonstrating how people are improving. There's a study in the University of Miami that's getting approval by the FDA. Another one in Colorado, then Texas. Israelis have shown 100% improvement in disease. I mean, all over the world, you're seeing different people with different types of viral conditions, including HIV, by the way. There's a study in England where they show the patient was completely healed. And so a lot of links that you can review for yourself and determine, the validity of that information, and then relate to yourself as well. In the US, we're looking at the right to try and a compassionate situation in patients that are critically ill and helping them. Again, I think the trials that are important are that there's other ones as well, so we can't ignore those. So basically, in conclusion about regenerative medicine, before I talk more about the COVID, the types of strategies or approaches that we have, it's first of all, it's cell therapy. It's biological and synthetic scaffolds, so that's the engineering part, where you create structure where cells can grow in. As I explained, you had the first engineered tissue or a skin implant. Then you have cell therapy and scaffolding. You have gene therapy. You've got reprogramming of cell and tissue types. And this is really exciting, where we with modifications can create that. The exosomes, which I haven't talked about yet, but exosomes are little particles, vesicles that are 40 to 100 nanometers in size, which are super small. And they have all these different kinds of cytokines, chemokines, that molecular products that actually function as the signals to the tissues. These exosomes, they also have specific markers on their surface so that they can recognize and home into the tissues and deliver their paracrine effect. You have the designer drugs, as I mentioned, basically where we look at cell cultures and figure out, recreate different disease conditions. It's brilliant how these guys do that, and so that you can then begin to look at how different types of drugs can ameliorate the conditions. And then finally, the delivery of soluble molecules. So far, therapeutics that are really acceptable are the blood dyscrasias. We do transfusions already, blood plasma. But the cell therapy worldwide is what we need to look at as well. I mean, there's many approved trials. I was part of an environment that had approval for using stem cells in a variety of different conditions with results that I'd like to share, but I'm not going to. Not on this platform, because again, this is what the FDA requires. The real challenge is that, we still don't know enough. We've got consumers that are quite desperate for solutions, willing to pay an arm and a leg, and then you have a lot of private clinics that aren't necessarily working out with the best of integrity. And so this is a problem. Sure, there are many that are doing a wonderful job, but this is why we do have the FDA to make sure that you're safe and that we're doing it in the right way. I'd like to think that the people that I work with, I work with an incredible company, which I'll mention in a moment, that has been in the tissue banking industry for the last 30 years. So they're very well integrated with regulation and understand what's right, and is doing a phenomenal job and has some amazing products. The kinds of products that we have, and you're going to want to know this, is that first, in therapeutics, you have what's called the cells. And they're from the Wharton's jelly. as I mentioned, from the umbilical cord. They could be also from the cord blood, and some of these cells are identical in the genetic material from the newborn. You've got the amnion. Some people are claiming that there are MSCs in there. I have to verify that because my understanding is that there really aren't that many at all. Epithelial cells perhaps, but really not MSCs. And then you have the placenta. And what we've discovered, through different studies is that it's the umbilical cord that has the highest yield. Umbilical cord has a higher yield than adipose tissue, and adipose has a much higher yield than bone marrow. And, studies have used bone marrow, but they've expanded these cells, which means they've cultured these cells so that they have enough cells. But again, per the FDA, is that these cells must undergo minimal manipulation. So expansion is, in effect, a type of manipulation. So you've got exosomes, as I mentioned. These are these little vesicles, 40 to 100 nanometers inside, with all these, all the goodness, as I like to call it. That's a scientific term, that delivers these molecules and substances to the tissues that then induce the stem cells to do what it is that they need to do. So the processing is really important, as I mentioned, because there's different ways to process it. And so a lot of people talk about the numbers of cells. I get befuddled when somebody comes up to me and says, well, I've got 200, 500 billion cells in exosomes. But then when you go down and delve into the science, is that to get that kind of number, then you have to go through a number of cycles. And then the more cycles you do of replication, what happens then is that you also lose the potency. So again, it's not about the number of cells so much. We always have to look at the actual therapeutic efficacy and benefit to really appreciate the value of the actual product. So one of the things that I get asked all the time is the quality assurance. How safe is it? And so one of the reasons I picked New Life Regenerative Medicine, just an incredible company. Vicki Mansavage is the CEO of this company. She's been in this industry for the last 30 years, quite a pioneer and a visionary as to what's possible. And they have the full gamut of different products, which I think is brilliant. Many of the companies, and some of them very good, by the way, just don't have the caliber of products that New Life has. But what I like about them is their absolute intense commitment to quality. And again, I think it's important whenever we are looking at different companies is what is that integrity? Because you're dependent on them doing the right thing, and do not want to get in trouble at any time with any kind of patients. I think what's important too is one of the big things that people are concerned about is transmission of disease. To the best of my knowledge, no one's ever has a disease transmission throughout the world. So I'm not just talking about New Life, but all cell therapies. So the quality assurance is pretty extensive. They go through complete medical, sexual, and social history, the physical exams, all sorts of other things, histories of transmission of disease, malignancies. They look at serology results. They look at HIV, hepatitis, syphilis, West Nile virus. So they look at a whole panel of different things that I think is really important. A side note, by the way, exosomes, and this is actually showed in the laboratory, just a whole gamut of effects that exosomes have with the products that they have, like the BMP5, it's not going to be much, but the BMP5, which stimulates the bone growth. So you have the SCF, which is responsible for stem cell and melanocyte growth. You've got the IL-6 responsible for macrophage activation. So all these different elements are key to understand what they are, and so you can also verify for yourself the value in it. I think in today's world with the technology and information that we have out there, it's a lot easier to verify this information because your doctor may not have all of it. that's again one thing that I'm hoping to change for doctors out there with the program that I have, which it's really exciting because it's really delved into the mindset of winners, the business tools of success in any industry, and then the science and integrity of regenerative medicine, so that you've got a great handle on the three critical components of any business. Even if it's not for regenerative medicine, it's for your own business. And I've been doing 20 years of research on this. And so I've had the privilege and honor to be working with some brilliant people over the years. Yeah, my practice, and now that I'm talking about that too, is that, it's all about harnessing the potential of the body to restore form and function. It's always been like that. As a plastic and reconstructive surgeon, I just didn't want to resort to plastic surgery. I mean, it's cool. Plastos means to change. And so, I didn't stop with just the physical transformation. The reason my podcast, as an example, and all the training and coaching and energy healing that I've gone through, realizing that it's important for us to tap into what I call the three intelligences: the brain, the body, and then the energy intelligence. And that's what I've been doing for the last 20 years. It's what I call smart medicine. I'm not just treating symptoms. What are all the other things? My first approach to any patient that I have, okay, let's see what we can do non-surgically. Just talked to a patient yesterday and says, hey, let's see if we can do this non-surgically, but finally decide, okay, we have to go through with it anyhow. When most people would, jump to surgery. Again, surgery is a big deal. But we live in interesting times. And as I mentioned earlier, it's exciting for all of us because it's absolutely dramatically going to change our lives. And this whole pandemic, if you want to flip it over and see the benefits of it, I mean, it's a horrible thing that's happening. Hopefully, we over-predicted what was going to happen, and it seems it might go that way, by the way. The pandemic in 2009, just in the United States, I think there were 60,000 people who perished out of 40 million people who got infected. So we're not even at those numbers, by the way. But it's still a scary disease, and it's important for us to take the appropriate action. But why is it an interesting time? Well, we begin with the wellness revolution, the intense desire to find out how do we optimize the body, which is great. And I've been working with a program called Optimize Me by Brian Johnson. Love the guy, love the guy, what he teaches. And it's really about optimizing your lifestyle and how to change your physiology to maximize it, but also using your brain. Then the second revolution is the regenerative medicine revolution. Really, again, as I call it, tapping into that body intelligence. How do you harness that potential of the body? And for me, even though that they're allogeneic cells, so these are cells coming from somebody else, they're still human. And they're still delivering a dynamic approach that we don't even can begin to understand how that works in tapping into the potential of the body to fix itself, to regenerate itself, to replace cells, and all these things. And so, my vision actually of the future doctor is that we won't be performing surgeries, we won't be performing or prescribing drugs. And together with the biotechnology revolution, right, which is not only a diagnostic, but the energy technology that can change and shift things. I mean, the three combined, I mean, it will be in Star Trek in the next 30 years. And I'm super excited about that. So I have about 10 minutes, if that, less than that, on the coronavirus, and I think it's relevant here. So just so we understand, the coronavirus, it's a common cold virus. Also the SARS, MERS were part of this group of viruses based on what it looks like. It's like a crown. As I mentioned, it's a bat origin. The rat's for the bubonic plague, and the bat is basically considered a flying rat. And out of Wuhan, that's where the evidence, and I'll let you decide how true that is. But there's a spike glycoprotein on the virus, and it's important to understand what that means. It's that spike glycoprotein connects with different cells in the body, specifically FS1 cell, the ACE2 cell, the DPP4 cell, and the CEA-CAM receptor. I'm sorry, S1 receptor, ACE2 receptor, DPP4 receptor, and the CAM receptors. Now, there are a lot of ACE2 receptors in the lungs, and this is why there's a propensity of the virus to go into the lungs. Also because, that's the entry into the human body. And so the ACE2 receptor is on the respiratory epithelium, so on the lining of it. And what happens is that once the virus gets incorporated into the cell, it creates what's called a biofilm. Then it expands and it ruptures the cell, and then you get what's called a cytokine storm. And so that's why, we need to not use exosomes, because exosomes deliver all those cytokines and could potentially make things worse. But that's just my opinion. I don't know how accurate that is. But the cytokine storm then specifically causes hyperinflammation and rapid damage to the lung, and the lungs fill up with fluid, and then people just can't breathe. And so that's why all the studies right now are looking to diminish that rupture of the biofilm, and I think that's where the anti-malaria drug is showing promise. Then you have the NK, or the natural killer cells, which are those lymphocytes, Hariri and Cellularity and Demantis are using, so that they can recognize these viruses and knock them out. And then you have the MSC cells, the mesenchymal stromal cells, or signaling cells, if we go with Kaplan, which then reduce the immune response and can also shut down that cytokine storm. And so, again, it's important for us to appreciate, there's this right to try, compassionate, to utilize what I believe the FDA would prefer is the umbilical cord cells. So there are, like I said, a variety of different studies out there on COVID with some pretty amazing results. And there's promise. I think it's relevant that we assume a lot of responsibility and integrity in how it is that we then pursue these trials or treat patients if you're a private clinic, and making sure that all the proper analyses are being considered so that we know that this works. The real challenge that we have right now is that the models, to me at least, are still somewhat unclear as to what is going to happen. There's talk about, well, all this lockdown, is that actually the best approach? Again, I'm no expert on that. There's such a thing called herd immunity. So at some point, is everyone going to get infected? And so what happens then? We've got an economy that's struggling. And so, people do need to go back to work. Certainly people that are very affluent and they can afford not to work for a period of time, but most of the world doesn't have that luxury. And so we've got to be smart as to what it is that we can do. And then also be smart in our preparation if there is a second wave. There is a suggestion that using the MSCs can provide an immunity for 3 to 5 months. Again, this needs to be verified. Once again, persons at risk, may benefit from these kinds of therapeutic options. The big concern, of course, as I mentioned, is that there could be a second wave in the fall. Again, not here to predict models. I don't understand the models. I think we just have to be really smart and understand, okay, what is our own responsibility in all of this? How is it that we can best implement? We've got to do the research. We've got to verify things. And ultimately, truthfully, as a coach, I coach many different people, from teenagers to billionaires. And, this is that opportunity to also revisit, our purpose on this planet, our responsibility in maintaining this planet. I did a recent podcast about the healing earth, and it just seems that the natural environment is enjoying the lack of pollution and all other things. Dolphins and whales are showing up where they weren't before. And so again, it's this opportunity that we can begin to reflect inward and see how we can become better humans in this planet, living in this planet as guests, as some people will say. what for me ultimately is the most exciting part is smart medicine. And for me, smart medicine is basically harnessing the potential of the body to restore form and function. It's what I've been saying for the last 20 years. Tried to implement functional medicine 20 years ago, but the science hadn't evolved enough. And so I stuck with other things. But now back in regenerative medicine, I've been doing that for the last 10 years with approved clinical environments and trials. And more importantly for me, my passion is to support doctors and patients with the ability to navigate this evolving field with confidence and the ability to make smart decisions for their future health, reducing the burden of disease, and enhancing the quality of life and our lifespan. I'm Dr. Bart Rademaker, Prescription for Your Transformation, real people, real conversations, and real success. And follow that link that's on the podcast to my page. Want more information, happy to give that to you. And again, I'm committed to also honor the FDA because they're the guides for us to make sure we do the right thing. I'll be back soon. Thank you very much, and talk to you later.
Thanks for joining us on another episode of Dr. Rademaker's Prescription for Your Transformation. Continue the path towards discovering your own authentic genius by tuning in next time for more real conversations with real people.